Okay, well, I can already smell it from here. So these little bluish guys, they have quite an odor, and that odor is anise. Or a chemical version of it. It's really intense, and these are anise funnel cap, Clitocybe odora. Odora, Latin for odor. So, oh yeah, I mean, really intense and nice odor, and it really helps identifying them. When they're younger, they're even more blue, and they're losing a bit of their blueness, bluishness when maturing. And interestingly, we've been doing here now for 4 years our funga study, flora, fauna, funga. Funga is the collective of all mushrooms. And with the Puget Sound Mycological Society. And we've been going out since 2016. We try to be here every second week during mushroom season. And then we try to do an inventory, what's out here. And for example, we haven't found this mushroom yet, which is around here in the Pacific Northwest, but we didn't find it. And I checked the list when I came across these guys a couple days ago, and we don't have it yet. So for our inventory, what we would do, we would get out our cell phones, and where's the cell phone? That's the usual process. Where is the phone? And then we would go onto iNaturalist, where we upload the data of what we find. So iNaturalist would give us the exact coordinations of the place. We would say make a new observation, and then we take pictures which shows the top of the mushroom and under the cap, so we can see all the characteristics. Take a picture, and that picture then will go online. Then we give it a name, and we also will take notes, like in this case, strong odor of anise or anise. And if there's color changes, tastes, all these things you only get when a mushroom is fresh is important to take. And often we would also use better photographic, better cameras to take good pictures. Cell phones are quite all right, but have their limitations. And then later on, we dry the samples at 135 degrees so that the DNA doesn't get destroyed. And then we keep the dry samples and submit them for DNA analysis. So, and that then can tell us if we have sometimes a new species or a new location for a known species, or okay, this is what grows everywhere around here, and there has been plenty of documentation even on the DNA level. But we already found some things, some new stuff within our 500 collections, and also new distribution area, like mushrooms that weren't known to exist on the West Coast showed up here. So this is part of the Puget Sound Mycological Society's Bridle Trail Funga study. And there's not a single study in the world about one forest or one location and the fungal inventory where you don't find new stuff. The curve slows down a little bit, but always keeps growing. And even mushrooms show up 50 years later, you know, where, wait a minute, have they been underground, the mycelium, all the time? Nobody knows. So really interesting aspect, and very helpful, and something badly needed. And all of this is citizen science. So we are not professional mycologists. We dedicate our time to do that, volunteer work, and we do learn a lot doing that kind of work. And actually in Europe in the 20th century, I think 60 or 70% of all new species, mushroom species, were described by lay mycologists. So it's a big tradition in mycology that many people, specialists, have other jobs while doing mycology as an obsession, I'd rather say.
So don't you collect a specimen and put it in the Burke Museum too?
Yes. So, well, at the moment, since we still take samples out for DNA and then microscopy and so on, we keep the specimens, but they will then be deposited in the Burke Museum, which is an important part because these mushrooms need to be accessible in a fungarium, so that if researchers who work on a specific small aspect, you know, like Clitocybe funnel caps, but the group around odora or so, they know exactly where to find it, and then they can work on it and ask for the specimens that can be exchanged between fungaria or herbaria.
Why do they call that one? Why is it a funnel cap?
Oh, the funnel cap. Clitocybe, which is Greek for funnel cap. Clito is the funnel and cybe is the cap in Attic Greek. Unfortunately, mycology is riddled with Greek terminology because I guess the botanists already used up so much of the Latin that they were forced, darn it, you know, this is already a plant, this is a plant too. So they used a lot of Greek, which is often much harder to associate than Latin. But so the funnel cap it's not as pronounced in this mushroom, but the gills will go down and it makes like a funnel shape. So some of its close relatives are much more funnel shaped, but the gills are decurrent. It means they go down the stem like a current of water, right? Which is an important criteria for many Clitocybe, but not all. So as things go. And that's the great thing now about DNA. DNA sorts these things in a certain way and helps us to decide which characteristics are really of evolutionary significance. Where does a mushroom belong in the tree of life? Before it's all a human decision. We focus on this aspect versus that aspect. And there was always discussion in the community. No, you got to look at the gills. No, look at the spores. Look at this, that. What really decides what is related? And DNA doesn't give us really that much of a choice anymore. Okay, they are related. Now, what do they share? That's the important traits.
But when you are identifying them after the forays, don't you look at them under a microscope, look at the spores and try to figure out what they are?
So that's definitely the classical approach is microscopy. Well, we key them out, but these days with the image revolution, we use much more images, and that gets us much closer. But then, of course, we go into the descriptions, compare these the traits, and there's some people are mightily upset doing these things without the traditional approach of lots of microscopy. And to do that, to do good mycology, that is definitely still essential. But citizen science, we can go pretty far without that. And if you have DNA on top, and the DNA, the names that are given is the DNA name is backed up by the first collection which got that name, then, you know, we can just go by that. But so there's definitely a shortcoming, and some in the mycology community are very upset of that kind of superficial approach without a, you know, description of every tiny detail. I say, hey, well, we take the pictures, we taste them, we smell them. So it's good, it's not perfect, but we get much more information much faster, and we have so much more information about species distribution. We can collect now probably in 1 year nationwide what took decades before, because it's so many more people. And yes, some of the information is there's lots of mistakes out on these citizen science projects. So it takes an expert to make the distinction what's right, what's wrong, or what gets close. But it's in the system, and then we can work with it.